Welcome back to Mighty Microbe channel. Today we are back with another cycle, that is citric acid cycle, or we also called as Krebs cycle or TCA cycle. So friends, it is also known as Krebs cycle or TCA cycle, that is tricarboxylic acid cycle. So this cycle, it was first introduced by Sir Hans Krebs, and this citric acid cycle, it is aerobic metabolic pathway, and the enzymes involved in citric acid cycle are associated with mitochondria in eukaryotes, that is fungus, algae, protozoa, plant, animal, and cytoplasmic membrane in prokaryotes, that is bacteria. So this cycle, it is universal cycle for aerobic metabolism, and this cycle, it is a series of reaction in mitochondria that brings about the complete oxidation of acetyl CoA to carbon dioxide, and it liberates hydrogen equivalent, which ultimately forms water. So this cycle, it consists of total 8 enzyme catalyzed reaction. So start with the explanation about this TCA cycle. So the first is condensation, where this oxaloacetate, it reacts with acetyl CoA and water to yield citrate, and in the presence of citrate synthetase enzyme. After that, there is a isomerization of the citrate into isocitrate. So this citrate, it is isomerized to isocitrate through an intermediary formation, that is cis-aconitase, aconitate. This isomerization, it takes place in two stages. The first one is dehydration of citrate to cis-aconitate, and the second is rehydration of cis-aconitate to isocitrate. So here it results in interchange of H plus and OH minus ion. This both the reaction, it is catalyzed by enzyme cis-aconitase. This aconitase catalyze removal of water from either citrate or isocitrate to form intermediate, that is cis-aconitate. And this aconitase catalyze the addition of water molecule to double bond of cis-aconitate by adding OH to either carbon. After that, the next step is oxidative decarboxylation of isocitrate. Here, isocitrate, it is oxidatively decarboxylated to oxaloacetate through an intermediary formation, that is oxalosuccinate. Again, this reaction, it takes place in two stages. The first one is dehydration of isocitrate to oxalosuccinate, and the second is decarboxylation of oxalosuccinate to alpha ketoglutarate. Here NAD or NADP, it is required as an electron acceptor. This both the reaction, this isocitrate to oxalosuccinate to alpha ketoglutarate, this reaction, it is catalyzed by enzyme isocitrate dehydrogenase. After that, the next step is oxidative decarboxylation of alpha ketoglutarate. Here, this alpha ketoglutarate, it is oxidatively decarboxylated to form succinyl CoA by the enzyme alpha ketoglutarate dehydrogenase. This reaction it is carried out in 5 steps. Is of succinyl CoA into succinate. This succinyl CoA it undergoes the cleavage of thiester bond, which is accompanied by phosphorylation of GDP to GTP. Here the enzyme catalyzing this reaction is succinyl CoA synthase. After that, the next step is dehydrogenation of succinate to fumarate. Here, succinate is dehydrogenated to fumarate with the help of enzyme succinate dehydrogenase. Here, FAD it function as a hydrogen acceptor in this reaction. Then the next is of fumarate to malate. Here there is a hydration of fumarate to malate with the help of enzyme fumarase. Fine. So after that, the next is conversion of malate to oxaloacetate. Here dehydrogenation takes place in the presence of enzyme malase. Here malate it is converted into oxaloacetate with the help of enzyme malase. So now in the next point, here detailed notes are given. How is the condensation of acetyl CoA with oxaloacetate, then isomerization of citrate into isocitrate, decarboxylation of isocitrate, then oxidative decarboxylation of alpha ketoglutarate. After that, conversion of succinyl CoA into succinate, dehydrogenation of succinate to fumarate, hydration of fumarate to malate, and lastly dehydrogenation of malate to oxaloacetate. So here the detailed explanation it is given how is the conversion. So see here, condensation of acetyl CoA with oxaloacetate. As this oxaloacetate it reacts with acetyl CoA and water to yield see here the reaction is given. After that, isomerization of citrate into isocitrate. So see, citrate it is isomerized to isocitrate through an intermediary group, that is cis-aconitate. Then after that, the next is oxidative decarboxylation of isocitrate. So this isocitrate it is oxidatively decarbolized to alpha ketoglutarate through an intermediary formation, that is oxalosuccinate. After that, the next is oxidative decarboxylation of alpha ketoglutarate. Here alpha ketoglutarate it is decarboxylated to form succinyl CoA with the help of enzyme alpha ketoglutarate dehydrogenase. Next is conversion of succinyl CoA into succinate. So this succinyl CoA it undergoes cleavage of thiester bond, fine, and it is converted into succinate with the help of enzyme succinyl CoA. And next is dehydrogenation of succinate to fumarate.